I, Dr. Nidhi Garg, Assistant Professor from Deshbandhu College, University of Delhi, welcome you all to EPG Pathshala. Today we will be discussing about Module 24B, which is Model Organisms in Genetics, particularly the zebrafish and the muscular, from the subject Molecular Biology. Now, after studying this module, you shall have a basic understanding of the vertebrate model organisms and their significance, the history of the use of the model organisms. You will know about the role of model organisms in the study of genetics. Also, you will know about the two most commonly chosen model organisms, that is the musculus and the zebrafish. You will also have an understanding of the specific roles of these model organisms and their extensive usability as non-human model systems. Now, in this module, we will be discussing about the two model systems like the zebrafish and the musculus. For understanding majority of biological phenomena, many non-human organisms, better known as the model organisms, have been resorted to. The major thought behind the use of model organisms was to gain insight into the working processes within the higher animals, and true to that, the use of model organisms did in fact provide great insights which kind of revolutionized the fields of molecular biology, genetics, developmental biology, evolutionary biology, to name a few. Now, the use of model organisms has been particularly of great benefit in studying human diseases, especially where experimentation needs prior and much difficult to get and ethical clearings. The use of these organisms reduces the risk of any undue or non-specific harm to humans, and these organisms are chosen when they have a certain level of taxonomic concordance with the humans. Now, although the model organisms have been greatly useful in such studies, but they don't provide any guaranteed effect in humans. But many of the drugs, treatment regimes and cures have been developed in or with the guidance of these model organisms. Now, the predictive, isomorphic and homologous are the three categories of the disease model systems. The predictive model predicts the human diseases, majorly the features of it. Those model organisms that share similar symptoms and treatments are isomorphic model organisms, while the model organisms that have the kind of highest level of similarity of causes, symptoms and treatment regimes as the humans having the same disease are called the homologous model systems. The model organisms are therefore chosen with great care depending upon the need and the level of experimentation required. Next we'll talk of the blast from the past on the use of model organisms. The use of model organisms in scientific research has been documented to ancient Greece, to as early as the times of Aristotle, when he conducted some of his experiments on live animals. Several examples of such significance followed thereby. The demonstration of germ theory in sheep using anthrax bacteria by Louis Pasteur in the 1800s, the use of guinea pigs by Anton von Lavoisier to prove that respiration is indeed a process of combustion was another example. The model organisms in a way have been central and of utmost importance to most of the important achievements in the field of modern medical science by providing great insights into the field of say biochemistry, neuroscience, human physiology, physiology of infectious diseases. Now, the use of fruit fly, Adia Drosophila melanogaster by Thomas Hunt Morgan initiated a kind of revolution, and it has become one of the most studied and the most widely used model organism. This actually fueled the change of biology into a solid experimental science. Now, during the time the use of Drosophila was spreading far and wide, the popularity of mouse model system was also gaining momentum, and the results were of great significance, especially in cases where inbred stains were to be produced. Since then, mouse model has gained a significant status as a widely used model organism and has been instrumental to several significant discoveries made in this as well as the last century. Now, the major contributors to the popularity of the mouse models were William Ernest Cassel and Abby Lathrop. Many significant examples were also from the field of immunology, like the Emil von Behring towards the late 19th century, successfully demonstrated the effects of an isolated diphtheria toxin on guinea pigs. This further led to bigger and more significant inventions such as development of vaccination against diphtheria in animals and finally in humans. This discovery has been instrumental in the reduction in the opposition towards using animals as model systems. As a result, it increased the resilience on model organisms. Now, subsequent research using animal models in the early 20th century has led to several important results, such as the discovery of insulin by John MacLeod. The consequences of using such animal models for research have been far-reaching and all-encompassing. The benefited fields were vaccination, development of antibiotics, organ transplantations, etc. While many diseases have been completely debilitated, animal models are still being used widely with the aim of solving yet unresolved medical problems such as AIDS, Alzheimer's, spinal cord injuries, multiple sclerosis, etc. First, we'll talk about the zebrafish, whose scientific name is Danio rerio. In this figure, the adult zebrafish is seen with dark blue stripes along the length of its body. Now, the zebrafish is a tropical native fish of the Southeast Asia with an average size of about 2.5 cm to 4 cm. The adult has dark blue stripes on its body along its length. The fish was first considered as a model organism in around 1960s. The following are few of its features that make these model organisms a useful model in research. First is its short life cycle. The sexual dimorphism is easily visible by distinguishing external features in male and female. Zebra. Male is slender with a pink or yellow tinge, whereas the female appears fat as they carry eggs in the belly. Mating is triggered in daylight and the development is quite fast. Its one-day developmental changes can be kind of compared to a month changes in case of humans. The figure 2 shows the adult zebrafish display sexual dimorphism, with the male being slender with a pink or yellowish tinge, whereas the female appears fat as they carry eggs in the belly. The generation time is about like 3 months, with ovulation and spawning occurring in presence of the male fish. Spawning can happen in 2 or 3 days interval. The fertilized egg turns transparent and is very useful as a model organism for development. The division is very rapid. Thereafter, within 36 hours of fertilization, all major organs start appearing. Like almost all of the yolk is consumed in 72 hours and a larva with head, body and tail is formed. In a month's time, the adult fish reaches sexual maturity. Figure 3. The figure shows the life cycle of zebrafish. It has transparent embryo and larval stage. The adult fish, as I said, has blue stripes along its body. The other important feature is easy in maintenance and quick development. The animal is very easy to maintain as they require simple natural conditions for propagation. The adults develop in groups like shoals, so require kind of less space. It is smaller in size and easy handling makes it economically better model organism than even mice. Then the external development with transparent stages is another benefit. The development of zebrafish takes place externally, which makes it an ideal model organism. A boon to this is the transparent nature of the embryos and the larval stages. The effect of any gene manipulation can be easily detected by the see-through body of the organism. The internal structures are visualized by use of light microscope. Thus, the use of harmful invasive imaging techniques can be totally avoided. In this figure, the zebrafish embryos are showing different stages of development. All stages can be easily visualized because of its transparent nature. In figure 5, the larval period in a zebrafish is around 72 hours to 30 days. The body is transparent to study the larval period development. Now, large number of offsprings is of course another benefit of using it. Research work on any organism requires hundreds of offsprings in a short interval of time. Because of this reason, too, zebrafish is a good model organism, as each female zebrafish can produce around 200 to 300 of embryos in weekly intervals, which is quite a significant number. Also, its complete genome has been sequenced, and was completely sequenced in the year 2013. It has some 26,247 protein coding genes. Now, since the genome is complete and of high quality, it has been possible to induce mutations in over 14,000 genes in the zebrafish. Also, the zebrafish shows several similarities to the human genome. Now, being closer to mammals in the evolutionary line, zebrafish share a high degree of genetic similarity with humans. Say around 70% of the sequence and proteins encoded are quite similar to human species, and this has led to several breakthroughs in studying human diseases. It has been known that some 84% of the genes in humans that are disease related share some degree of similarity with the zebrafish genes. Coming on to anatomy, the human anatomy is similar to zebrafish as both being vertebrates. The organs and tissues, especially muscles, blood, kidney and eye of zebrafish has been used to study and compare the anatomy features of human system. The other important benefit is the genetic manipulation of zebrafish embryos have highly permeable skin and can easily absorb chemicals dissolved in water. The chemical mutagens are added to the water where the embryos are kind of developing. These mutagens then they cause mutation and its effect can be like visualized through their transparent body. Talking about their research work in case of genetics, forward genetics mutagenesis includes introducing random mutations and screening the phenotype for the genes of interest. This method has been very successful in zebrafish as fishes are maintained in a controlled environment and other variations can be avoided. Many such gene variants that hold similarity to human system have been investigated. Talking about heart muscle regeneration, the zebrafish displays high power of regeneration in the heart muscles. It has been observed that the fish regenerates the heart muscles if they are removed experimentally. Scientists have been working for a long time to study this mechanism as it can prove very beneficial for the human heart ailment. It can be used for the repairing of muscles that are affected in case of cardiac failure or attack. Now, they can be genetically bioengineered biosensors. Scientists have created transgenic zebrafishes which have estrogen responsive elements that control the expression of green fluorescent proteins or the GFP. This help in detection of environmental pollutants that have high levels of estrogens. Now upon exposure, the ERE are activated that in turn regulate the formation of GFPs. Now in this figure, the University of Exeter's engineered zebrafish, it has GFP expression under the responsive elements. The fish illuminates, due to the production of GFP, the fish illuminates and serves as an excellent biosensor. Many compounds like the beta estradiol, cadmium chloride, zeaerol, estradiol, ethyl stilbestrol and bisphenol A have been tested and has produced excellent results. This fish is also sold as a pet and is known as a glowfish. It is safe and easy detection method. Now in this figure, the genetically engineered zebrafish having a transgene of GFE under control of ERE is being used as a biosensor. Talking about research work, other kind of things, the understanding the mechanism of muscle dystrophy, heart physiology, drug development against skin cancer are a few examples of research aspects in the zebrafish. Next, we will take up the mouse, that is the mus musculus, which is the most preferred of all model organism when studying human system and is widely used in the clinical research. It is the most preferred of all model organisms when studying human system and is widely used in clinical research. It is basically termed as the laboratory mouse, which is used as a model organism. Now, it is descended from the house mouse, mus musculus. There are three subspecies, that is the western house mouse, which is the mus musculus domesticus, the eastern house mouse, the mus musculus musculus, and the Southeast Asian mouse, the mus musculus castaneus. The laboratory mouse is kind of a hybrid with a mosaic genome from the above mentioned three subspecies. There is a difference in the coat color of the wild type and the lab strains. The wild type has brown coat, while the lab strains can be albino or sometimes have black fur. It is generally small with like 7 to 11 centimeters in length and an average body weight of 20-25 grams. Talking about its life cycle and the short lifespan, the generation time in this model system is relatively short. It takes around 10 weeks time for a mouse to be able to give birth again. The another advantage is its short lifespan of 1 year, which is contributed in aging related studies in human. It has been known that 1 year of mouse is like equivalent to 30 years of a man's development. The female has a 4 to 6 day estrus period. A copulation plug develops after fertilization and the gestation period lasts for about 19 to 21 days. Each time, 3 to 14 young ones are delivered. Now, the female can give birth to around 5 to 10 such litters in a year. Breeding is a continuous process and the mouse breeds throughout the year, leading to quick multiplication in its population. Now, the figure 9 shows the short life cycle of the musculus. The other important feature is the cost-effective handling and maintenance. These organisms are very cost-effective in maintenance. The handling is quite easy as they are small, multiply quickly and are able to give birth every almost like 3 weeks. The female is ready to mate on the same day of giving birth. Talking about its genome sequence, the complete genome sequence is available and was published in the year 2002. It has 3.5 million base pairs in length and has 23,000 protein coding genes. It is approximately same number as present in humans. However, in humans the genes are distributed on 40 chromosomes. In figure 10, the mouse having 23,000 protein coding genes are shown distributed on the 40 different chromosomes. Now, talking about its similarity to the human system, the number of protein coding genes in mouse and human is similar. Also, many diseases like atherosclerosis and hypertension are being researched in mice also possess similar genes that are responsive for these diseases. Not only the genetic makeup is similar, mice also shares similarity in anatomy and physiological aspect with the human system. They can develop cancer and are known to suffer from diabetes like diseases. Because of this reason, it has been extensively studied in relation to human diseases. They also help in understanding the human immune system, the endocrine system, the nervous system as well as the skeletal system. Talking about figure 11, the mouse and the human brain possess many similarities that has been used to study brain mechanisms and diseases. Talking about gene manipulation, it has been possible to induce diseases like cystic fibrosis and Alzheimer's in mice, which generally do not occur naturally in this organism. It is induced by genetic manipulation and by providing required environmental conditions. Talking about immunodeficient mice, scientists have been able to grow human tissues, both normal and disease, in mice whose immune system has been made dysfunctional. These are called the immunodeficient mice and have been hugely useful in studying cancer and AIDS development in humans. Talking about the mouse model systems, the mice models that are unique strains produced by selective inbreeding to get desired results are highly useful in researching upon diseases like cancer, diabetes, Huntington's, etc. Behaviors like anxiety, aggressiveness, alcoholism and even drug addiction is also being studied in mice. Another type of these mice are the transgenic and the knockout mice. Now, in recent years, mice has been manipulated by inserting a foreign gene to create a transgenic model. Also, genes can be deleted with the help of techniques like homologous recombination to produce the knockout mice. These two models have been a breakthrough in studying the role of different genes in the human genome. The figure 12 illustrates the development of a transgenic mouse by the process of microinjection. The Jackson Laboratory is a special lab that is dedicated in developing different mouse models. It is situated in Bar Harbor, Maine, and has played a crucial role in areas that include cancer development and aging, immune system and blood disorders, neurological and sensory disorders, along with certain metabolic diseases. They have special repository which builds up a kind of a consortium of sequenced genomic data of different strains of mice. It has a collection of 2,700 strains, either in the form of like breeding mice or as DNA samples. A few of these mouse models used in different diseases are listed below. Like they've been used in Down syndrome, a model named TS65DN mouse mimics trisomy 21 that's responsible for Down syndrome in humans. The mouse is known to exhibit many of the behavioral patterns in learning associated with learning and physiological defects that are generally observed in case of humans who suffer from this syndrome. Cystic fibrosis. A knockout mouse model has helped to understand that cystic fibrosis occurs when a small change in gene blocks a channel that regulates salts and water movement in and out of a cell. That leads to like failure of clearing of bacteria that infects the lung and the mucus accumulation and the lung disease. Talking about cancer, a knockout model P53 was created in which TRP53 tumor suppressor gene was disabled and gave insights in studying various cancers including the lymphomas and osteosarcomas. Simulation for studying genetic disorders and new drug development are its other advantages as a model organism. Talking about the human growth hormone, the transgenic mice having the human growth hormone releasing factor has been a great breakthrough in studying many growth related disorders in humans. This model displays increase like in somatic growth, but they generally turn infertile somehow. So now let us summarize what we have talked about in this module. Zebrafish has dark blue stripes on its body along its length. It displays sexual dimorphism and has a short lifestyle. It is easy to maintain and has transparent developing stages. The large number of offspring and genome similarity to humans is a boon for the scientists. It has been used in genetic manipulation for studying heart muscle regeneration, understanding mechanisms of muscular dystrophy and cancer drug development. Mus musculus is the most preferred of all model organisms when studying human system and widely used in clinical research. The generation time of this model system is again relatively short and easy maintenance and cost effectiveness is seen along with the easy handling. The genome is completely sequenced again with a size of 3.5 million base pairs and an equivalent 23,000 protein coding genes. The mouse bears great similarity with the human system in terms of genetics, physiology and anatomy. This has helped in developing mouse models to get insights in human diseases, new drugs and genetic therapy. Thank you all of you. I thank all of you and I hope that my module will help you in understanding these two very beautiful model organisms.